Hello students, welcome to EPG Pathshala program. I am Dr. Shoaib Lukman from the Council of Scientific and Industrial Research, Central Institute of Medicinal and Aromatic Plants, Lucknow. Today, I am going to talk about the next part of the types of lipid from the paper Lipid Metabolism. The objective for today's lecture is to discuss another important type of lipid which is called as sterols, and among sterols, how many different kinds of sterols are present in living organism, what are their significance, and how they perform their function. Among the sterols, cholesterol is the main sterol present in the tissues of vertebrates. The word cholesterol is derived from the Greek word chol, which means bile. The cholesterol is the carbon 27 alcohol which is present in high amount in adrenals, gallstone, liver, and nervous tissues. Brain is principally rich in cholesterol, covering 1/4 of the total free cholesterol available in the vertebrate cadaver. Cyclopentanoperhydrophenanthrene ring, or also called as styrene ring, is a carbon structure of cholesterol, and it is the first sterol isolated by Pelletier de la Salle in 1758. This isolation occurs from the gallstone. Later, in 1815, Chevreul isolated cholesterine from the unsaponifiable fraction of animal fats. Continued further, Reinitzer in 1888 proposed the accurate formula of cholesterol, but exact steric depiction and structural information came only after the work of Wieland, who later won Nobel Prize in Chemistry in 1927. The work of Wieland includes the establishment of bile acid and related substances, followed by Window's work, who got Nobel Prize in 1928 for establishing a connection between the sterols with the vitamins. Gallo and Young in 1936 have voted that steroids, which is chemically linked to cholesterol, in 1913, Steenberg proposed the central role of cholesterol in atherogenesis. So, we are discussing different types of lipids, and these types include the simple lipids, complex lipid, and derived lipids. The simple lipids, including the fats, the oils, and the waxes, have already been explained in the previous modules. In the complex lipids, which includes the phospholipids, glycolipids, and proteolipids, has been discussed herein. Phospholipids are phosphorus containing lipids, whereas glycolipids are lipids containing saccharides or the carbohydrates. When lipids are amalgamated with proteins, it forms a complex mixture of lipids which is called as proteolipids. So in this module we are discussing about different types of glycolipids, and it has been continued from the previous modules. So among the type of the lipids, and in complex lipid especially, sterol constitutes an important role. And among the sterols, cholesterol, which is one of the most abundant form of sterols in animals, play a vital role in the physiology of the animals as well as humans. In animal cell membrane, the concentration of cholesterol is usually high, ranging from 30 to 50% in erythrocytes, and as high as 80% in the membrane of ocular lens. Accordingly, cholesterol has numerous tasks in membranes, varying from the control of phase behavior, offering mechanical strength, to precursor of hormones and vitamins, and in scheming, raft arrangement, and membrane protein activity. Minor modifications of the side chain led to the replacement of cholesterol by sterols such as campesterol, beta-cytosterol at the cellular level. In the femoral gland of Acanthodactylus boschianus, which is a male lizard, cholesterol is present in abundance and has been used as a scent blotching pheromone to ascertain the dominance. In addition, cholesterol can also silhouette ester connections with secreted polypeptide and signaling molecules encoded by the hedgehog gene family that function in some molding action among metazoan development and evolvement. The sponges signify the richest source of peculiar sterols, that is cholesterol, but bearing 1 to 3 extra carbon atoms at C24 with unusual features as quaternary alkyl groups, acetylenes, alleines, cycloprenes, and cyclopropane rings. In class Desmospongi, 24-isopropyl cholesterol with its analog unsaturated at C22 and C23 is abundant and characteristic of Neoproterozoic era sediments and is the primeval proof of fossil record for animals, but it is absent in the Eumeta zone. The presence of cholesterol is now largely accepted in higher plants, detected in vegetable oil in a small proportion, that too 5% of total sterols. A high content of cholesterol is present and reported in the oil of camelina, a biodiesel plant. Nevertheless, numerous studies have shown the presence of cholesterol in chloroplast and the existence of cholesterol in pollens and in the leaves of higher plant. Cholesterol is the only sterol of Lawrencea paniculata species and also the foremost in the most of the rhodophyceae algae. Lanosterol is another type of complex lipid and the common precursor in the synthesis of cholesterol. Lanosterol is also found as a foremost component of the unsaponifiable fraction of full fat, that is lanolin, which constitute about 15% of the lipids. Gemmata of Scuriglobus, which is a bacterium of Planktomycetes, is capable of producing lanosterol and its unusual isomer named parchiol. The derivatives of Lanosterol have been reported in methanotropic bacteria, 4-methylcholestein 8,14,24-diene 3-beta-ol is an abundant derivative first reported from the Methylococcus capsulatus species, and later on it is reported in other closely resembling bacteria. Compounds with lanostein and have been successfully isolated from a mushroom, Ganoderma lucidum. The presence of these sterols have led to the utilization of mushroom for curing diabetes, cardiovascular disease, bronchitis, gastritis, hepatitis, hypertension, hypercholesterolemia, and related pathologies in traditional Chinese and Japanese medicine. 7-dehydrocholesterol is another complex type of lipids that is present in animal tissues, which on ultraviolet exposure gets converted to cholecalciferol, that is vitamin D3. The source of ultraviolet light is principally the sun. 24-dehydrocholesterol, which is also known as desmosterol, is an intermediate product between the lanosterol and cholesterol, and it is associated with the myelination processes with a high level noticed in the brain of young offsprings, but absent in adults. Gorgosterol is another type of sterol, a complex lipid that was revealed by Bergman from the coral-like animals. Belonging to zooxanthellae, and they are the intracellular photosynthetic dinoflagellate symbionts. The structure contains cyclopentano perhydrophenanthrene ring, like that of cholesterol. Another important structure which is present in dinoflagellate belonging to subtype Sterols are Dinosterols. These Dinosterols are 4-alpha 23-24 trimethyl, 5-alpha cholest 22E in 3-beta all, and this is considered to be an unserving biomarker present in dinoflagellates. In animal tissues, 7-dehydrocholesterol is present in minute amount, which get transformed to cholecalciferol, which is also known as vitamin D3 on UV exposure. Another important 24-dehydrocholesterol, which is called as desmosterol, is an intermediate product between cholesterol and Lanosterol, and this is associated with the myelination process with a very high echelon, and noticed in the brain of young animals, and no desmosterol is found in adults. In astrocytes and spermatozoa of mammalian cells, this is an abundant membrane component, whereas in red algae 20T2 dehydrocholesterol and desmosterol are present in high concentration. Desmosterolosis is one of the severe disorder with a cognitive impairment and developmental defect in humans, where the desmosterol fails to get converted to cholesterol. So conversion of Lanosterol to desmosterol was possible, but from desmosterol further on, cholesterol is not formed, and that result in a very severe cognitive impairment. In 1943, Bergman revealed gorgosterol from coral-like animals, and these are intracellular photosynthetic dinoflagellates, symbionts belonging to zooxanthellae. The inventive structure abides a cyclopropane ring in the side chain and a very unusual C23 methyl group, refurbishing the interest in marine sterols. Dinosterol has been found as an unserving biomarker present in dinoflagellates. Oxysterol is another type of sterol, a subtype of complex lipid, is present in brain tissues as 24S hydroxycholesterol. It is a type of oxysterol which is primarily synthesized in the brain. In 1953, it was noticed that in horse brain and thereafter named as cerebrosterol, 24S hydroxycholesterol has been proposed as a biochemical marker for Alzheimer's disease, protecting the formation of beta-amyloid peptide found in plaques. The other aspects of oxysterols have also been reported and reviewed. In animal kingdom, the starfishes have oxysterols, which is being illustrated by several hydroxylation reaction with no complement. From a far eastern starfish, which is zoologically named as Henricea leviscula, the structure of 15-alpha cholesterol hexanol is reported, and that what makes this oxysterols a unique and specific complex lipid of starfish. The chlorinated cholesterol is another subtype of the sterol belonging to the complex lipids, reported as alpha chlorohydrins or synonymously called as 6-beta chlorocholesterol, 3-beta 5-alpha diol, and other chlorinated products are generated by myeloperoxidase, hydrogen peroxide and chlorine system. After reaction with the double bond of the cholesterol second ring, these alpha chlorohydrins are relatively firm and have been found valuable as a lipoproteins marker. Oxidatively damaged by the triggered phagocytes, which secretes myeloperoxidase enzyme. These chlorohydrins were also produced in low density lipoprotein and membrane of the cell, ensuing exposure to myeloperoxidase system or hypochlorous acid. The chlorohydrin's formation in cells might be unruly to membranes, resulting in the lysis of the cell death. These chlorohydrins are also used as the biomarkers for oxidative damage associated with monocyte or neutrophil activation. So, today in this module, we have learned about the different types of sterol, the presence of these different sterols in different types of organism, and the significance of these sterols and their role, starting from cholesterol to 7-dehydrocholesterol to 22-deoxycholesterol, desmosterol, dinosterol, Lanosterol, and Oxysterols. Each of these sterols has some uniqueness, some specialized functions, and because of this, they have their importance. In the forthcoming module, we will continue with this complex type of lipids to discuss more about the different types of lipids present in organisms. Thank you.